Good morning to everyone who has gathered here. On this glorious day, I feel privileged to welcome everyone here for the final session of the International Webinar Series BioFIESTA 2023, organized by the Department of Biotechnology, Sri Shankara College, in collaboration with the Internal Quality Assurance Cell and with financial support from the SMART scheme. God, our Creator, has imbued our minds and personalities with tremendous potential, strength, and ability. Prayer enables us to access and develop these abilities. Now, with the blessings of God Almighty, let's unleash our full potential. Let's bow our heads and immerse ourselves in the holiness of God Almighty. I invite Jesli for the prayer.
विश्वम्भरं विश्वस्रजं विश्वयोनि अनश्वरम् विश्वस्तमात्मजं वन्दे विश्वोत्कर्ष प्रचोदितम् यत्पापमकरोत् पूर्वं नराणामातिमो नरः कैवल्यं ततुर्युक्तं वन्दे विश्वम्भरम्
Before moving into the depth of the scientific session, I would like to invite Hanna Elias for the welcome speech.
A warm welcome to one and all gathered here. The galaxy of intellectuals, Your Excellency invited eminent speaker, guests, professors, and dear friends. It is a matter of great pleasure and pride for me to welcome you all to the final session of the International Webinar Series BioFIESTA 2023, organized by the Department of Biotechnology, Sri Shankara College, in association with the Internal Quality Assurance Cell and with the financial support of SMART Scheme. Before moving into the depth of the scientific session, let me get done with my duty to welcome you all. Firstly, I would like to thank the experts for taking their precious time out and making to this event. Secondly, I would want to thank my audience for being present here today. Before we begin with our today's session, please allow me to deliver a short speech in order to warm up everyone regarding today's scientific session. Today's webinar scientific session mainly focuses on the potential of bioengineered 3D models in biomedical research. Over the past decade, advances in biomedical and tissue engineering technologies such as cell culture techniques, biomaterials, and biofabrication have driven increasingly widespread use of 3D cell culture platforms and subsequently the use of organoids in a variety of research endeavors. Given the 3D nature of these organoid systems and the frequent inclusion of extracellular matrix components, these constructs typically have more physiologically accurate cell-to-cell and cell-to-matrix interactions than traditional 2D cell cultures. As a result, 3D organoids have the capability to serve as superior model systems than their 2D counterparts. 3D cell culture has emerged as a promising method for the generation of organoids that could serve as a model to study various disease mechanisms or the toxicity of new drugs. Organoids are highly heterogeneous 3D structures that exhibit typical tissue architecture. So, in today's scientific session, we are focusing on a very relevant title, the potential of bioengineered 3D models in biomedical research. It is my honor to welcome our resource person of the day, Dr. Binulal Nelson Sathy, Assistant Professor and Ramalingaswami Fellow, Amrita Center for Nanoscience and Molecular Medicine, Kochi. Welcome you, sir.
Thank you.
On this occasion, I have immense pleasure in extending a warm welcome to all the faculty members who worked so hard to make this a successful program. Welcome you all, dear teachers. Last but not least, I welcome all the faculties, guests, professors, students who are here to attend the webinar. On behalf of the Department of Biotechnology, Sri Shankara College, I welcome you all. Thank you.
Thank you, Hannah. Next, I would like to invite Fiona to introduce today's distinguished speaker. We are extremely fortunate to have Dr. Binulal Sathy with us today.
It is a matter of great pleasure and pride for me to welcome our guest of honor, the eminent speaker of today's event, Dr. Binulal Sathy, Assistant Professor and Ramalingaswami Fellow, Amrita Center for Nanosciences and Molecular Medicine, a distinguished scientist, an excellent professor, a seasoned scholar, and one of the eminent scientists in the field of bioengineering. He has completed his Bachelor of Science in Life Sciences from Kerala University and his Master of Science in Biotechnology from Manoன்மணியம் Sundaranar University, India. After his post-graduation, he worked as a research assistant at the Institute of Biomedical Science, Academia Sinica, Taiwan. During the period 2012-2013, he performed as a visiting scholar for a collaborative research program with Rice University, USA under USISTEF Indo-US Visiting Scholar Fellowship. He received a junior research fellowship from the Department of Science and Technology, Government of India during his postdoctoral studies on bioengineering at the Institute for Nanosciences and Molecular Medicine, Amrita Vishwavidyapeetham, India. Then he served as a postdoctoral research fellow at the Trinity Center for Bioengineering, Biomedical Science Institute, Trinity College, Dublin, Ireland, from 2013 to 2016. He has over 20 peer-reviewed research publications and 2 book chapters to his credit. His research interest is in understanding the fundamentals of immunomodulatory and regenerative properties in adult stem cells and bioengineered matrices for functional tissue regeneration. His work also focused on the translational aspects of stem cell products and cell-derived matrices for regenerative medicine. He is a recipient of International Travel Grant from the Indian Council of Medical Research 2019. International Travel Grant from the World Council of Biomechanics Travel Grant Travel Award 2018. He received Ramalingaswami Re-Entry Fellowship from the Department of Biotechnology 2017. He represented India at the Asia Nanotech Camp 2010, held in Singapore. On behalf of Department of Biotechnology, Sri Shankara College, Kaladi, I am honored and indeed proud to invite Dr. Binulal Sathy to the scientific session. Welcome, sir.
Thank you.
Thank you, Hannah. It is now time to begin our scientific session. Following the presentation, we will have time for an interactive session where you can ask questions using the chat box. Participants, please ensure that your audio is muted and your video is turned off. Please feel free to ask any questions you may have. We will set aside time at the end of the presentation to respond to your questions. Success is no accident. It is hard work, perseverance, learning, studying, sacrifice, and most of all, love for what you're doing or learning to do. It is a matter of great pleasure and pride for me to welcome Dr. Binulal Sathy, Assistant Professor, Amrita Center for Nanosciences and Molecular Medicine, Kochi. Sir, please.
Hey, good morning, all. Thank you very much for. Can you hear me? Thank you very much for that nice introduction. I mean, can I get a response? Am I audible?
Yes,
Okay.
yes, sir. We can
All
hear
right. All right. First of all, I'm delighted to be part of this BioFiesta 2023, organized by the Department of Biotechnology and International Quality Assurance Cell, Sri Shankara College, Kaladi. And I extend my thanks to the organizing committee to invite me to be part of this great event. And today, I'm delighted to know that I have a group of young minds who are working in the area of biotechnology and microbiology because they are the future. Their thoughts and their commitments and their expertise are necessary for the country, and their innovations are absolute necessary for us, for our country to grow. So, with this small background, um, I'm happy to share, happy to discuss with you the topic which we selected, which is from bench to bedside, the potential of bioengineered 3D models in biomedical research. Um, so I'll be sharing the outline of my today's discussion. And if you want, you can stop me at any time and you can ask me questions, or as the you planned, you can ask the questions at the end of the session also. Um, so because we are all, I mean, there is a big similarity between me and all of the participants in this conference today, or who are all listening to me today, because we are all working in the area of something related to biomedical research. Some of you may change your expertise after you graduate from your college to other fields such as agricultural field or something else, but majority of them are working in the area of biomedical research. And we are all doing this. We are all using our precious time, effort, and energy to contribute something to this field. So when we do this one, from our bench, and finally the success means when it is translated and when it is reaching into the necessary community, that's the success, that's the translation. But when we do that, there are a lot of hurdles. So then I'll be introducing you the need for surviving that difficult path which is commonly called as the valley of death. Then I'll be introducing you how monolayer cell culture systems are being used and what all were their advantages, and is there any limitations associated with the monolayer systems, monolayer cell culture systems. Then I'll be just teaching you the cells which are highly used in the biomedical research, especially before for a proof of concept research, are highly influenced by a lot of factors other than the conventional media and the culture conditions, such as including the cell shape and stiffness, etc. So I'll be just introducing that to you. And then I'll be trying to convince you that there need to be a paradigm shift in the in vitro systems from the conventional monolayer cell culture systems. Thereafter, I'll be giving you examples of some of these systems, bioengineered in vitro systems that can be used for in vitro studies, and then we'll be concluding this today's discussion with the limitations of whatever I'm proposing as the three-dimensional systems. And of course, I would be concluding that there. And as I said, what means this surviving the valley of death? So any research, any innovations all starts from somebody like us who got basic training in the area, in the biotechnology, biochemistry, microbiology, and other fundamental sciences. The After understanding the necessity, we will be using our thinking power to see what we can do in this specific area. In our case, in the case of biomedical research, there is a small change where the necessity comes from the bedside. In our center, what happens is the necessity comes from the bedside. That is, the clinicians will pose the challenges which they are facing, or they will be telling us what they need, or they will literally they would be asking, is there anything possible which we can do in this area? Then, as researchers, scientists, what we our commitment is to go back to the bench and try to come out with solutions to that. And the interesting fact is the study is not stopping there. When we come out with some kind of an innovation or a solution, it is our responsibility to see whether it can finally reach into somewhere back into the bedside. So it is kind of a cycle, bedside to bench to bedside. So that is the complete cycle. I'm not saying that everyone will be able to do this successfully, but constantly all of us who are working in this field are trying to do that. So when we do this, especially you all may be aware that biomedical research is an expensive business. And we know that many of the life saving procedures, drugs, and commonly used drugs, everything came into existence after going through this rigorous process of innovation to the translation. So it has multiple phases from the basic science to translation to human, translation to patients, translation to practice, and then to the translation to the community. So mainly we are all working in the basic science to the translation to the human. So I'll be focusing much more in that area. So think about any drug which costs us around 100 rupees today. What would be the actual cost of producing that particular drug? I would say I'm not going to give you any number, but I would say maybe 1% of what they are charging us to a maximum of 10%. That's what I would say. Then why they are taking this much money from us? Why the companies are charging this much money? Because many of the innovations, even though we have good amount of data to say that it is working, when we try to take it into a manufacturing process, and then when we do a trial in humans, finally ultimately we are making it to do use it in the humans, most of them will fail, most of them. So finally, if a company is investing billions and billions of money, majority of their innovations and their money will be lost, but one or two would be successful. So all the loss and their benefits, their profit, everything will be charged in the successful product. They will be adding all this into the successful product. That's why all these medicines we have to pay extra. So is there any way to make this innovation in an affordable way? That's the big question which I'm throwing to you. Think about it. What could be the best option? So the valley of death means from the academia to the industry, we will, when we, you know that from the cell in vitro systems, we will go to the preclinical systems, large animal systems, and then you go to the phase 1 trial, phase 2 trial, phase 3, phase 4. So most of them, even after the large animal studies, they largely fail. They won't show a significant effect or some kind of a side effect when it reaches to the human. Then it can't be used in the human. So that is the value of death. Many of the innovations go like this one. So what is the way to overcome this? Is there a way to overcome this challenge? What is the goal? To make affordable products in the area of biomedical research. Okay. So I would say this can be made more affordable by use of human engineered tissue models. We all know that, including me and you, we are using cell culture systems, monolayer cell culture systems, to do all our research to identify is there that compound is working well, that compound has any effect, whether that is useful, whether it's an anti-cancer agent, this, that, etc. But I completely agree. Many of our fundamental understanding on the human system, human cells, and their molecular biology, cell biology, all came out into the textbook knowledge through the monolayer cell culture systems. All right. However, we should appreciate that cell behavior in normal as well as in pathogenic condition is a lot different. Mostly we will be using some cell culture systems, some monolayer cell lines or primary cells. However, in the actual individual or in a patient, cells are not remaining as a single monolayer systems. This is a big difference. So we have to consider that. Now, the advantage of the monolayer system is, I agree, they are easy and convenient. The results are reproducible. More importantly, you can take it into the microscope and you can watch, you can see what is happening, whether the cell morphology, whether the cells are dying in response to whatever treatment you are doing, is all possible. Now, because as I said, now I have to make you convinced these cells in the monolayer system may not be the same as what it is in their original place. Okay, why I wanted to prove this one? Because most of our innovations are initially identified from the monolayer systems, and we assume that they are all gonna be successful when we try into the in vivo systems. So to make any innovation affordable, if a product is gonna be failing, it has to fail as early as possible. If we know that the system is working minimally, we should be able to identify in the initial screening step, not when we reach into a phase 1 clinical trial. How is it possible? It is possible only by giving a more relevant system. That's my point. So now I will try to convince you that the cell can be the cell shape and matrix stiffness can affect on the cell behavior. So here there is a study which we did. This is an example where we made a three-dimensional system, a particular system where a particular cell, for example, here I have selected mesenchymal stem cell, they can remain in two different shapes in the same gel. You understood? So two different shape means one is just round cell, one is the spread cell. Okay, the same, and then it is a three-dimensional system where they can remain in two different shapes, just changing the shapes. And I can keep this system in two different stiffnesses. One is soft, which is around 4.9 kilopascal, and the other one is stiff, which is around double the stiffness, for example, 13.4 kilopascal. Remember, we have two shapes and two stiffnesses. Okay, and well, everything is good. The cell is the shape we can get. This is an example of how the shape can be, and in all the systems our cells are viable. The green indicates viable cells and red dot indicates dead cells. This is a calcine staining. This staining is in a three-dimensional gel, and now there is not much of a difference between any of these things. Now, but is there any difference then? Any difference whether the cells are influenced by any other factors? Okay, let's see. We have these cells seated on top of a soft hydrogel, and we have seeded the same cells on top of a stiff hydrogel. I said we have two hydrogel systems, but components are the same. We engineered in a way that the components are not changed, but just cross-linking is induced more so that I get a stiff hydrogel versus a soft one. Soft one is let's just like soft like a flimsy gel. The other one is a little bit stiff. Now you see that the two cells you see under the actin staining heading, you can see that the shape of the cells are totally different. Same cell, only difference which you did is just the stiffness of your substrate is changed. Okay, I'm proving that my concept is ready or my argument is ready, is true, because the cell behave according to the environment. Okay, now I have more interesting result here. Yep, it's a signaling molecule, you know, right? That the translocation of this transcription factor, YAP, from the cytosol to the nucleus can control the associated gene expression. Here, you can see that when these cells are on top of a soft hydrogel, the YAP remain in the cytosol. It is not going into the nucleus. That's the first row. End of it, you can see that. The first one is the nucleus, then I have the YAP stained, and then the DAC image. And when you merge, you can see that most of the YAP is still in the outside. If it is in the inside, the color will change. Now, interestingly, you see, when the same cell on a stiff surface, the YAP is going into the nucleus. That's why the color is changing. So this is telling you what? This is telling us that even though the cell is the same, the media is the same, only by changing the substrate, the molecular mechanisms in through which the cell behave is changing. Now think about, you are just doing some experiment with this cell by putting in a tissue culture plate. Tissue culture plate is a very stiff one. It is not same as that of the stiffness or softness in the body. So are we looking into different molecular mechanisms? Possibly, yes. That's my point. So if you are convinced with this one, we'll move to the next one. Now, in the literature, you will see that in a stiff surface, the YAP will go inside. We also proved it. But in a soft, it will remain in the outside. We also saw that. Now, Now this is a story when the cells were on top of a hydrogel. Is that correct? Now, what will happen if we put these cells inside the hydrogel, which is more like the in vivo system? Inside the hydrogel, we made it, we mixed the gel with the cells, and we again attain more or less the same. The spread as well as stiff and round, soft. Now we have four systems: round cells in a soft hydrogel, round cells in a stiff hydrogel, then spread cells in a soft hydrogel, spread cells in a stiff hydrogel. All these cells are inside the gel. Now what we observed, again we did whether the yap is going inside or not. Regardless of the stiffness or the shape, none of the yap has reached the nucleus. So again, I'm proving you that the environment itself can change the behavior. So now think about, you are identifying an important mechanism or a response of to a particular drug using a cell which is on a soft substrate. But when it changes, when this environment changes when you try in the human trial, your results won't be observed in in the situation. What it means? So if this is not going to work, we have to eliminate it in the beginning. That's the only way to minimize the cost and make affordable products. Okay. Now again, I have this one to show you that compared to soft and stiff, the alkaline phosphate positivity in these cells are significantly different. Alkaline phosphatase positivity is significantly different. The only difference which you have to remember is, in one, cells are in a soft hydrogel, the other one it is a stiff hydrogel. Components of the soft and stiff are the same, cell is also the same. So the stiffness is making a big difference here also. So I convinced you that. Now again we continued that, and you can see that all the genes associated with adipogenic differentiation and osteogenic differentiation are expressed differently between these two systems. I think I am not going to the details of how the experiment is done, but this is basic qPCR data showing that fold differences in the associated genes, which we checked in two different systems, one versus the other. And it's convincingly tell you that the white bar indicates soft, the black bar indicates stiff. It is different. At least take that as a point. Okay. Now I proved that the stiffness and the cell shape can influence the behavior of the cells. Okay. Now why that is important? Because in your monolayer systems, you are not considering the cell shape or the stiffness. You are just plating it there, culturing them as a monolayer system, and you are studying it. And many of the times it may be relevant. I'm not saying that it is irrelevant in all the aspects. It may be relevant, but whether we are mimicking the environment similar to that what is possible in the final translational setting, that is also very important. And now I'm trying to convince you, or I'm giving you evidences to say that structural dimensions and architecture of the matrix can also influence cells. Here, as an example, I've selected macrophages. Macrophages, as you know, these are human macrophages. They can exist in two polarization stages, M1 and M2. You would have studied it. Maybe M1 is an inflammatory one, M2 is an anti-inflammatory one. That means when there is a wound happens, there will be an inflammatory environment initially, which actually triggers these macrophages to produce necessary cytokines to attract all the inflammatory cells to there. Eventually, these macrophages will polarize to an M2, which will produce cytokines and chemokines necessary for more tissue regeneration at that site. That's the background there. So macrophages are important. So here, see, I have four systems here. One is same polymer. That means the first three are polycaprolactam. It's a synthetic polymer, which is made into nanoscale dimensions, which is nano around 0.284 plus or minus 10 micrometer. A point, no, a 0.1 micron, and a micro, which is around 10 micron, And then micro, I have a different dimension, which is around 300 micron. It is 3D printed. As a control, I have a decellularized bone. So 4 systems there. Decellularized bone means natural extracellular matrix. Okay. So just look at these scanning electron microscopic images. Macrophages are the same. Macrophages are same, isolated from the Buffy coat samples collected from the hospital side. So see how the cells behave. The first one, they are like more spread. Second one, when you gave only the structure is slightly different, their behavior is slightly different. When you gave a bigger fiber, they are more like round. And in the natural extracellular matrix, it is slightly, totally different. So here, when you check the inflammatory cytokines and anti-inflammatory cytokine, for example, IL-10 is an anti-inflammatory cytokine. So you see there is a significant difference in the level of the cytokines produced from different systems. What is different here? Only the structural dimensions. Maybe associated with associated stiffness there, which I haven't measured, but obvious one is the structural dimension, same polymer, same cells, same media. Now the inflammatory cytokine level is different. So I hope that is convincing to you. Now that was structural dimension. Now what we did is we checked whether the alignment of cells, alignment of these structural dimensions. You can see in the nano, they are all parallelly aligned. In the micro also, they are parallelly aligned. Micro 2 also parallelly aligned. And the control is just control, like the natural extracellular matrix. You can see, okay, there is a difference, the way cells respond. Okay, in the same, just because they are different in the size and in the alignment, it is influencing how the cell is behaving. Ultimately, any disease which you are going to talk about, ultimately it is starting with the cells, and there is where you are trying to reach. So with these two results, I assume that I convinced you at least to the point is that the monolayer systems may not be necessarily mimicking the in vivo like conditions. You may be knowing it. Now, what is other options we have? Of course, we have, and we were doing it with animal models. Okay, we can start it with, you know, fishes, or much more. Even we know that there are insects, worms. These are all animal models. But consider something which we are using. It's a, if you still remember your 12th grade zoology, you would be knowing that fishes, amphibians, reptiles, aves, and mammals. And in the mammals, we are the most evolved one. So we are trying to understand the disease, which is in the most evolved, more highly complex organism, by studying it in a much simpler form of organism. Yes, we may need these in vivo systems to understand certain level of complexities, but can we consider something which is working in a mice or a rat or a rabbit or in a sheep can be the same as what is happening in human? I won't agree with it now, but many of these things are necessary to understand fundamental mechanisms. I agree. So we have two extremes here. I'm not saying without monolayer culture or animals, we can go into a different level. No, we need monolayer culture systems to have initial some kind of a screening. We need animal systems to understand it. But before we go into much more expensive studies, we have to ensure whether this is gonna be failing, or is what is the percentage of success for this particular innovation which we are trying to do. There comes in the potential of engineered tissues. Okay, tissue engineering or engineered tissues. Now, I'm inviting all of you to the world of tissue engineering, and using with the latest advances in the bioengineering field, we can recreate in vivo like conditions in vitro. Okay, these are beautiful. The first one is not my data, it's a human brain which is recreated by organoid technology. That means if you have a stem cell, now we know how to induce them, how to control them in a three-dimensional fashion. So what is an organoid? It's a miniature version of the organ of interest, which can represent at least, which can mimic at least one of the functions of this respective organ. So that is possible. A lot of studies are happening in the organoid. So there, my point is, a study which is done at the beginning in the TRL, technology readiness level 1 to 2, using a brain organoid may be much more relevant, and the data which is coming out from that will be much more relevant than a study which is conducted using just a monolayer culture of some of the glial cells or neurons on some other cells which is there in the brain. Now, in our lab, this is a picture from our lab. This is we are because I'm interested into the bioengineering aspects. We will come to that in the bone marrow, and we were able to recreate it. It's just a representative picture. So I think I introduced this tissue engineering area to you. Basically, you can recreate in vivo like, I'm not saying in vivo condition, in vivo like environments in vitro. An in vivo study will be much more expensive than an in vitro study. So if you have in vitro systems which has capabilities to predict in vivo like results, it's going to revolutionize our the production cost, or the, I mean, that is the stepstone for the affordable, making these innovations affordable. Okay. So the ability to generate human tissues in vitro from stem cells, primary cells, has raised enormous expectations among the biomedical research community, patients, and general public. Now, I have to introduce some of the studies which I am doing, which include these developing bioengineered tissue models, ex vivo expansion of HSCs. These are the things which I am doing, growing large volumes of bone and musculoskeletal tissues. So these are generalization of the studies which is happening in my lab. So one of the very interesting tissue or an organ which I'm fascinated always is our bone marrow. You know that they are very crucial. They are the center for hematopoiesis, and more importantly, they are the center for many of the malignancy metastasis. For some reason, these cancer cells, when they are cancerous, they like to go into the bone marrow. That's one reason. Second one, hematopoiesis. Hematopoiesis, all our immune cells and blood cells are formed in this one. And bone as an organ, as well as the bone marrow, is always attracting to me. And uh, you can see that bone is the third most frequent sites of metastasis behind lung and liver. So there is something specific there. What I'm trying to say is, if I can recreate the bone marrow like environment in vitro, a lot of studies can be done to understand why there is metastasis towards this bone marrow. See, for example, prostate and breast cancer are responsible for the majority of the skeletal metastasis. One reason. And now I'm using this for as a engineered bone marrow, like a microenvironment for the expansion of HSCs. Hematopoietic stem cells, they are the stem cells responsible for all our blood cells, including and our immune cells. Now, you know that hematopoietic stem cell transplantation, which is largely known as bone marrow transplantation, is a life-saving procedure, especially after hematological malignancies or any disorders in the hematopoietic system. Now, um, haploidentical donors, matched donors or haploidentical donors are the only possibility. And for this transplantation, the patient has to wait to identify the donor, and there is an associated time delay. And most of the time it is, but think about a scenario where these stem cells are available like the blood bank. Just go to the bank and get it and treat the patient, and that will be a much faster recovery or much faster action which is happening. So, but what is the problem? But nobody will just come and donate their bone marrow just like that. Okay. So now we have to look into affordable sources or easily available sources. One of the easily available sources is the umbilical cord blood after delivery. This is a medical waste if they are not storing. Nowadays, there are some patients are or some people are interested to store it, even though that's not an approved process from the from our governing bodies, but they are storing it. Otherwise, it's just a medical waste. We can get 100 to 150, 120 ml of cord blood after the childbirth. So, and it has a good population of hematopoietic stem cells. But if you consider the number, that number is not enough for the transplantation. It would be good enough for a transplantation in an infant, maybe less than 5 kg. But a person with 60 kg, the number has to be that high. So that is not available. So only option is you have to expand this hematopoietic stem cells. So what we did is we were trying to see. So now scientists came out. Okay, they can expand these HSCs, but because they were also using the cell culture plate and they were throwing these HSCs inside and using a cocktail of cytokines and chemokines, the cells number they attain, but their function is lost. Because in the bone marrow, the functionality of the cell is highly controlled by the cytokines, chemokines, and the microenvironment, whatever is produced from the niche cells. We call it as bone marrow niche. Okay, so what in coming in, to summarize, what we did is we selected two platforms. One is a decellularized bone, and the other one is just like what I showed you, a synthetic polymer based matrix, and we engineered it in such a way that we can recreate that in vivo like niche using the stromal cells and endothelial cells. These are just a representation of it, and you can see more interesting images showing that. So we can have mesenchymal stem cell containing zone. You can see that these are a synthetic polymer where these cells are growing. Okay, growing, and this is a scanning electron microscopic image, and you see endothelial cells also. You can just grow into these strands. What it means that now you have a three-dimensional environment which is like the bone marrow. I'm not saying it is bone marrow environment. Now we have to see whether we can grow these hematopoietic stem cells isolated from the umbilical cord blood. If we can, whether they are more functional compared to a 2D expansion. Okay, so from here, I mean, I have one more picture to show that they are the cells. When you seed it with CD34 positive, this is a characteristic surface marker for your hematopoietic stem cell. When you seed them, these bone marrow niche cells um, are helping them to attach onto them, and these cells are behaving better. I mean, this is just a picture to show how this is after seeding the HSCC. These HSCs go and bind with these niche cells in the environment. Now, when you when you check I mean, this I have some quantitative data. This is I mean, this we recently published. You can look into it. And what we observed is compared to a 2D culture, okay, a 2D culture of any of the niche cells, when you culture these niche cells in the 3D, either using microfiber or any other system, the osteopondin, which is an important factor which will make this HSCs quiescent. Okay, that level is significantly low when you have this 3D culture. That will be helpful for expanding this HSCs for our purpose. And now you see, using that three-dimensional environment, we try to expand our HSCs, and you can see we can get up to 40-fold of expansion, and these are media conditions. Okay, different media conditions in a three-dimensional environment. You can just seed your niche cells and then you seed your HSCs, and you can get up to 40-fold. Okay, so now what that means, so if you have HSCs which can be sufficient for a person with 5 kg, if you expand them without losing its functionality, you can get around 40-fold. Maybe we are reaching there. Okay, so this has been published recently, and so that's about the bone marrow studies or the environment, three-dimensional environment we have created in the lab. Currently we are working, and interesting, I haven't included, we found that these HSCs which are cultured in these engineered niches are more stem like. They have more stemness characteristics compared to the 2D culture system. The second one which I'm want to tell you is the bioengineered bone for any kind of osteoporosis or infection. And bone is formed by two mechanisms which you might be knowing. One is intramembranous ossification. That means the stem cell will directly differentiate into osteoblast and bone will form. That's how our flat bones are formed, like the skull, etc. Now the other one is endochondral ossification. Endochondral ossification is nothing but that's how all our long bones, okay, all our long bones are formed. What happens there? Mesenchymal stem cell will condense. This happens in the embryonic development. Okay, the MSCs will condense. They will form cartilage first. They will undergo hypertrophy, and then slowly that will form bone. This is the background. So we followed it, and we can finally we demonstrated that we can recreate the bone, engineered bone, and these systems can be used as a substrate, as a model system to study osteoporosis or any kind of bone infection. I think I'm not going to the details of what exactly it is, may not be very relevant to you, but my point is you can see this micro CT data which will behave like bone, and he have alicerendered staining which is showing that is mineralized. Now, in many cases there are interfaces from the cartilage, bone, and then ligament. Even we can recreate that kind of a bone to ligament interfaces. Here you can see that interface how it is. Here beautifully you can see that mineralized one and then the ligament. This is the insertion site. So that's also possible. Okay. Now, second one is the osteoarthritis. We are all familiar with the osteoarthritis. What is osteoarthritis? Is nothing but our cartilage is not. Cartilage is, what is cartilage? Cartilage is the tissue which is helping the articulation. Any of the articular cartilage is nothing but the cartilage tissue present in the articulating joint. Articulating means moving joints. Everywhere there is cartilage. If this cartilage is not there, means we are stiff. We won't be able to move. It will be pain. So when we are young, energetic, this cartilage will be beautiful. Over the ages, the cartilage will undergo some kind of a damage. When damage cartilage, you have to remember this cartilage is one tissue which is avascular. Okay, the moment it is starting to degrade, the vasculature will come, nerves will come, and immediately pain. You won't be able to move. Okay, so and osteoarthritis is a big problem, and if a small lesion form, if it is not properly treated, then it will be finally ending up in a situation where the total knee replacement, it's a end process. It is the only option. But if there is a small lesion, if you can fill that with engineered cartilage, that is possible. Or we can study why this osteoarthritis is happening using the cartilage, engineered cartilage model. So we were successful in developing cartilage tissues on a dish. Now we have this one compared to putting chondrocytes into a plastic plate. You can have this one and study it. And the staining is nothing but it is showing the GAG content. Don't worry about these different columns. These are different compositions, but it is beautifully showing that it has a lot of GAG there and collagen there. Collagen. Picrosidase red is for collagen, and aldehyde fusion Alcian blue is for GAG content. And what is a characteristic for articular cartilage is the type 2 collagen. You check this one, the bottom row of the engineered cartilage, depending on the composition, you have the engineered tissue with articular cartilage like architecture and composition. Type 2 cartilage is there. This has also been published from our side. And then the next one is, of course, I said that this is useful for in vitro studies. So now we were trying to model prostate cancer metastasis to bone. Okay, now I convinced you to show that we can do bone. Okay, we can create models of bone, and we have three-dimensional environments where it can be a substrate for forming any tissues. That means including cancer. So we started into prostate cancer, and we have a specific gel which we created there. We call it as in situ gel forming a collagen based hydrogel, because you just make this one. It's a composition of collagen with alginate. That the beauty of this gel is it is remodelable. That means the cells can degrade, they can move around, and you don't need any specific cross-linking. So you just inject, it will polymerize, and you can see the SEM. It is like the collagen fibrils are forming there, right? And the cells are very much happy to live in this particular gel. They are all green. There is no red dot, saying that they are all viable. And so we have created, using this gel, we created this prostate cancer there. And also we created the bone organoid. I showed you this particular gel mixed with this MSCs and induce them to the bone. For example, BMP2, they will form bone organoid. We created it. Okay, and it is characterized and it is forming bone. And now we have the tumor which we created. This is an experimental model which we did. We created a three-dimensional tumor. That means you take the gel and put the prostate cancer cells isolated from the patient, or PC3. cell line which is available. So you have it, and you keep your bone organoid at a distance from this tumor. Okay, so I have a bone organoid containing slot here and a control without any bone organoid. And if it is a specific metastasis, what will happen? The tumor cells, prostate cancer cells, because we know that prostate cancer will metastasis to bone. If there is something happening in the bone organoid which is attracting to this, you will see a more specific migration towards the bone organoid, right? In the other side, it may not be there. So this is the experimental system. And here you can see that the prostate cancer cell in the gel, and you see day 0, day 1, they started coming out, day 5 and day 7. So they can come out. Now we from after understanding this, we went to the bioprinting thing and we bioprinted a platform where I can keep the bone organoid in the center. No, I think the prostate cancer in the center and bone organoids at different arms to see whether the cancer cell, hope you understood. So we can have bone organoids at 2 and 2 other thing. You can just keep the gel to see whether the prostate cancer cell have any preference to move towards the bone organoid containing one. This was printed in our lab. And then, for example, you can see here you have blank as well as bone organoid containing environment, the experimental system. Now so this big story in short, what we observed is after at day 7, there was, you see the DNA in the migration channel, and we checked how much DNA is there in the channel which is connecting the end pockets to the bone. For example, the first one at day 0 there is nothing, and this is a system where you have treated with this prostate cancer with any drug. You can see that in the when there is no bone organoid, nothing is moving. But in the case of bone organoid, you see from the metastatic cancer, a lot of cells are moving. It is there in the channel. See, it is reflecting as the high number of DNA. The DNA content is very high there. That means if you are the cells which is not treated, they can specifically move towards the bone. Now, when you treat it with docetaxel, the migration is substantially decreased. Okay, in the bioengineering perspective, what is important for me is we were successful in creating a system which can tell the investigator whether which drug you are not tested before you go into the patient. You check it here, maybe it is docetaxel, maybe in a different dosage, maybe in a combination. That can be done. So this is another example for the 3D system which we created here. I think I tried to give you examples of systems which we are working with, and the need for these 3D systems compared to the conventional 2D systems. Now, is there any challenges associated? Even if it is all fancy to say that you can just create it, the 3D system, and do all your experiments, and then you are going to be successful. You will be successfully able to reduce the cost. At this stage, till it is expensive, and there is lab to lab variations. That means if I created an organoid and somebody sitting in the US, when they create an organoid, for example, even the cartilage or the prostate cancer, on the or the hematopoietic, the bone marrow niche, there is a lab to lab variation. We haven't reached into a stage where there can be a consistent protocol to recreate. That's a challenge. And now there is no off the shelf availability. All these 3D systems, when you want to go into analyze something, there is no system which is all need to be customized. You can't just go and take it into the microscope if there is no fluorescence coming in, because it's an opaque thing, three-dimensional thing. It's not like your plate. And of course, there is expenses associated with the establishment of the engineered tissues. At this stage, it is expensive compared to your 2D systems. But if we establish this successfully, at least for those studies which are showing very good promise for translation, I highly recommend that this has to be tested in a three-dimensional system to properly understand or to predict their in vivo performance. If, as I said in the beginning, if this system is gonna fail, let it fail in the beginning, so that it will be less burden to the people. Finally, all the burden will be coming to the community, right? When we come out, a company come out with a product, they will charge everything what they lost, including and the expected profit in the new product which is coming. Who is going to buy it? Layman, right? Common people. So in that aspect, I think this is one option which we can go ahead uh, to identify what is the actual possibility of that system to be successful. Okay, and just to conclude, there is an urgent need for alternatives to monolayer cell culture systems for evaluating drugs and medical devices, and bioengineered tissues and organoids are the potential options in the place of monolayer culture of cells in tissue culture plastic. And these systems may produce data that can be relevant to human body. Personalized tissues and organoid development is possible, and it is the future. More investigations are necessary to produce engineered systems that can recapitulate in vivo like environment ex vivo. And this is my team. I have three PhD students working with me: Akhil, Hridya, and Niji. And Saptagiri was my master's student. He moved to Germany. And Aravind is in Bangalore now. These are new three master's students working with me. And now our center, just because I said a lot, our center is offering different programs in nanobiotechnology, nanoelectronics, and nanoengineering, and molecular medicine at undergraduate level, postgraduate level, and MTech level. And please look into our website if anybody who is even after masters, those who are interested in the technology programs, tech programs, they can choose it if they are interested. And finally, I want to acknowledge the funding agencies and of course giving us this fantastic infrastructure to conduct all these studies from Amrita Vishwavidyapitham and ultimately Amma. Thank you very much.
Thank you for providing such an informative session, sir. Now
You.
it's time for the interactive session, and I would like to invite everyone to come forward and ask their questions in the chat box.
Um, am I expected to just answer based on whatever is posted here, or it will be asked directly from somebody?
Sure,
I can see, yeah,
there is a question in the chat box.
yeah.
May I read them out?
No, I can read. Like I can see that biomedical applications of 3D bioprinting. Yeah, I mean 3D bioprinting. I would see it as one of the cutting edge latest advancements in the area of three-dimensional tissue regeneration. So you said it as 3D bioprinting. I think you should understand the difference between 3D printing and 3D bioprinting. 3D bioprinting, 3D printing is possible if you want to develop a model system, a human body, okay, any organ. If you have the digital information, you can 3D print it. Even in our center, it is possible because we have one of the latest one, the 3D bioplotter, or that's possible. A model system using a thermoplastic polymer you can, but bioprinting means printing a functional tissue. Biomedical applications for that will be, for example, we are using it as dot prints or of small organoids for cancer tissues. You mix it and you can get, for example, think about it. Any in vivo tissue is not a homologous, assembly of single type of cell, right? So if you want to study the spatial interaction between two layers of cells using bioprinting, it is possible. You put your first layer and suggest the printer to put the second type of cell at close proximity to the first one, it will do. That's the simplest example which I can give you. But the application will be on developing organoids or 3D in vitro systems at this stage for medical evaluations as model systems, etc. eventually, but the tissue engineering community is trying to take this 3D bioprinting to print functional organs. We haven't reached there yet, but that's the ultimate goal. That is the answer to Nikita. Then Elsa, so could you 3D models of cancer enhance? Of course, yes. 3D models, because I haven't included that here because one of the studies which we were, again, the prostate cancer, we were testing how it will respond to docetaxel treatment, just treatment. Okay, we had a 2D versus 3D, and we had a repeated dose versus a single dose. In all the conditions, the 3D systems were less responsive, showed less response to the drug. What it means, in 2D, the cells are all dead, or they are less active or less viable. But the same concentration of drug in a 3D system, the cells are kind of resistant to it. Maybe it is telling that is more relevant than the 2D evaluation. Maybe when you actually try it in a in vivo system, maybe the result which you would get would be similar to what you see in the 3D. That's one example, because we are just starting into the cancer models. The one model which we tested is prostate cancer. So I would say 3D models, bioengineered models, but it can't be just any cell putting into any gel and mix it up and make a 3D. No, it should be a relevant 3D system which is like the at least closely mimicking the in vivo tissue. Then it is a much advanced system to screen, drug screening. Because if you find a response in a 3D system, that is going to be a more relevant when you actually reach into the clinic, into the translational stage. You understood? Hope you understood my point. But what is happening is now the screening is done into 2D systems, and maybe you start with 1,000 molecules and you reach with 100, and this 100 they have to take into preclinical and clinical trials. No, preclinical and large animal studies. It's hugely expensive. Now, when you try that in the 3D models, you might, from the 100, you may come out with the 2 or 3. You take those 2 or 3 into the next stage. So in that way, that will be helpful. Potential of 3D bioengineered, I would say that was the previous was the answer to Elsa, and the next to Anamika. Anamika, a lot of studies are happening into brain organoids. I think that in the organoid area, brain organoids are one of the established one, and a lot of studies, because in the neurological things, it's all communication. The neuroscientists are experts in studying how communication is happening. Their communication between cells and between different layers are all important, and a lot of studies are focused in that area. So if they can do fundamentals of this, how the brain works using models, of course I am very sure that they can be used for studying the neurological disorders, but we are not doing anything at the stage here. That is to Anamika. Varada. At this stage, I would say there is no successfully printed 3D printed organ. That is the hope that eventually we will be able to, because see, I would say, I said that printing in vivo like tissues, okay? So when you consider tissue, it is assembly of different type of cells in different ways, including not only just cells and extracellular matrix, and most importantly vasculature and the nerves. So at the moment you can't incorporate vasculature and the nerves into any printed tissue, in my understanding. So I would say that 3D printing at the moment is only successful in developing. For example, in our hospital, what we do here is maybe a patient comes, an infant has a cardiological problem and he has to go for a cardiac surgery, some valve problem, and we have the three-dimensional data. What we can do is we can print the entire physical organ like the heart using just a polymer, not with cells. Now the surgeon can actually see through where they have to do the surgery, how to approach, how to cut, make the initial cut, how to repair. That can be practiced. That is happening now, but not as transplantation, in my understanding. The last one, Hannah is asking, brittle bone. I mean, uh, brittle bone is what you mean by brittle bone? I think, is that osteoporosis what you meant? Is it like just breaking the bone? If it is, can you, Hannah, can you just,
Yes, sir. Yes,
yeah,
sir. I meant osteoporosis.
osteoporosis. Yeah, osteoporosis is because, um, you know why osteoporosis happens, right? It is either osteogenesis is compromised, it's imbalance compared to the osteo bone resorption. The osteoclast activity is more than the bone formation. All right, or bone formation is
Yeah.
not happening in the osteoclastic area, so that's why it is just getting broken. But bioengineered bones will be helpful. For example, a piece of bone has to be replaced in that area. If it is in a localized area, bioengineered bone can be helpful. But osteoporosis is not affecting only in a localized site. It will be it is kind of a systemic problem. The entire bone will be kind of compromised, right? When as the disease progresses. If it is not something localized, bioengineering may not help. But bioengineering will help to recreate the osteoporotic bone in vitro, and you try all the possible drugs and identify some kind of a drug molecule which can be given to patients. Hope that satisfies your question. I mean, that answered your question.
Yeah, okay, sir.
Yeah. So when just now I want to know, when did you realize that you have an interest to 3D models? See, this was I'm into the biomedical research for the last 20 years. At least after the initial 3-4 years. At that stage, I mean, at that stage, it was just the beginning, because um, uh, in the biomedical research or in any aspect, you would see that if alternative options are there, you always go for it, right? At that stage, there were no other options. You can't just go and see. Think about any disease which you are. I mean, of course, we are concerned about human beings, and only after that it will be veterinary medicine, right? So something which is happening into human, we don't have the right to go and test something directly into the patient. You should understand this one. Anything, okay, in the simple way, because as a person who is working in the regenerative medicine, I will say, if you take some kind of a tissue cell from a patient, some bone marrow cells, you take it to the lab, and if you take it back to the patient, it is not permissible, it is not allowed, it is highly regulated. Okay, that means any disease you can't directly go and test it into humans. So that means what? You are left with only the other animals, which are, it is different species. Okay, and again, just going and testing it with some large animals, you have a lot of regulations like ethical clearance, and people will be, nowadays in India also, we are all animal lovers. So there is a problem. You can't just go and inject something into the. I mean, in olden days, even I know that in World War II and all, like, human beings were just used as they were. There was no regulation at that time. If the people or the scientists want to test it, they will capture a group of people and just inject. If they die, they die. That's all. Nowadays, it's not like that. It's highly regulated. So now we how is option is the animal models. In the animal models also, there are a lot of regulations. You have to, in our center, we have to first go for a scientific committee review, and only if the scientific committee approve, we can propose this into the animal ethics. Animal ethics has to get cleared, then only you can do, and there limited number of animals will be allowed. That also in the lowest in the evolutionary tree, but it's like, mice or rat. Only after you produce that data, you can go into larger animals like goat or something like that. So the point is, when you realize Okay, with the limited available resources, whatever innovation is taken to a company is taking it up. And when they do invest a lot of money into this, and when they reach into the clinic, they are failing. That's what the value of death. Many you just Google it and understand what is the percentage of the success of biomedical basic innovations. So my point is, we somebody is spending that much money and it's failing finally. So now who is going to invest? Nobody will invest means there is no progress. We won't have any kind of medicine. By realizing that was the turning point for me to go into the bioengineering and 3D models. Okay, but this is we are still in the infancy. But a lot of research is coming, a lot of data is coming into the literature suggesting that they are better to predict the therapeutic outcome or the final outcome. Simply as I showed you, you have 2D cancer, prostate cancer, and a 3D one. A given concentration is killing everything in 2D, okay, and in 3D it is not killing. What it tells, think about this, I used a drug. If it is a new molecule, you will think that, oh, my molecule will successfully kill all the cancer, and you will go to the next study, next study, next study, and finally when it into the clinic, you will see that there is no big differences with the existing drug versus your new molecule. So all the expenses and your effort, time, it's all will be compromised, right? At that time you will feel rejected or something like. So we should try to pick something. It's just like an early detection, just like, if your system is not functional, if it is something to reject, stop it, stop it. If it is something which you have to refine it as early as possible, not after the clinical trial. So in that way 3D models are going to be helpful. Is it possible to, I think that's answer to just now, and is it possible for the cells to change its genetic behavior after 3D printing and culturing? I would say the first one, 3D printing only recently came, and the culturing, I would say it is very subjective because it is, as I said, I was trying to expand HSCs, right? Now even though I successfully generate required number, I will need to clear all the genetic screening and mutations, possible mutations, what happened to that, before I go for approval from the regulatories. If I want to go to a clinical trial, but what it indicates, there are possibilities, especially think about a fibroblast in your body or in a epithelial cells in your body. It is sitting in a very soft place. You throw it into a tissue culture plate, which is highly stiff. It's a polypropylene, and the cells are going there, and I showed you enough data to say that the matrix can change its molecular ways in which the cells respond. So in that way there can be changes happening during the cell culture. Okay, culturing the cells, again it depends on how long you culture, how many times you passage them, how many times you treat it with a proteolytic enzyme, how gentle you are in handling the cells. Now the second part is in printing. Printing as such is not actually giving that kind of a stress to the cells, if I understand. The shear force and all in the 3D bioprinting is very minimal. So, but even though when I say very minimal, it is significantly higher compared to a cell just sitting without going for a 3D printing. But comparatively, it is minimal. But I am not ignoring the possibility of any kind of genetic alteration after printing. But it is very subjective. That means different type of cells will have different level of, many of them may not have any changes, maybe one or two might be very fragile cells. If you do, it might be something which need to be tested out. Hello. Yeah.
Thank you. I believe the
Yeah.
speaker answered almost all of the questions. It is now time to conclude the session. No duty is more urgent than that of returning thanks. The two words that will change your life are thank you. Like a candle that can light 1,000 more without shortening its own life. Appreciation is a gift that, when given, can set the whole world aglow. I invite Elsa for the vote of thanks.
It has been a great privilege to be a part of this wonderful event. On behalf of the Department of Biotechnology, I would like to express my heartfelt gratitude to our esteemed principal, Dr. Preeti Nair, the management of Adi Shankara Group of Institutions, and IQAC for their encouragement and strong support in the successful completion of the international webinar series BioFiesta 2023. On behalf of the Department of Biotechnology, I would like to express my heartfelt appreciation to our esteemed resource person, Dr. Binulal Nelson Sathy, for his very informative and interesting talk. The webinar provided an in-depth look at the significance of research in bioengineered 3D models in biomedical research and its various aspects. Thank you once again, sir, for your well-organized and knowledgeable presentation. My sincere gratitude to the department head, Mrs. Sujay Lakshmi KV, who handled the event throughout. I would also like to thank the organizing team, volunteers, guests, professors, and my dear friends who worked tirelessly to make this webinar a huge success. Finally, I would like to thank all of you present here with a wide round of applause, and thanks to all the participants who made this event a memorable one and for making the time to be with us today and helping this event make a grand success. Once again, thank you all.
Thank you. The
Thank you,
link to the
guys.
feedback form is provided in the chat box. The link will only be active for 1 hour following the session. Thank you for being such an amazing audience and for your cooperation. Thank you.
Thank you. All right.
Thank you, sir.
Thank you.